मेरे को देखते रहते हैं, मास्क पे बनता कुछ बनता।
Good morning, everyone.
Good morning, everyone. Panika sir, I am audible?
Yes, you are very much audible,
Yes,
but
sir.
the light seems to be a bit less. Your image is quite blurred, I suppose.
Image is blue.
The light there is not sufficient.
Good morning, sir.
Yeah, good morning. Good morning, everyone. Yeah, now it's better. Qureshi madam, now it is better.
Okay, sir. Thank you.
Yeah.
Karthik, am I audible? Hello, Karthik, unmute yourself. Unmute yourself. Am I audible?
Yes.
Sir, the honorable person for today's seminar, Dr. Siva Prasad Adhikari, has also joined us. And Adhikari sir, our principal, Dr. Vikas Domne, has also joined us.
Namaskar, sir.
Okay.
Mini echo येत आहे तिकडे जा.
Adhikari sir, the principal of our college, Dr. Vikas Domne sir.
Good morning, sir.
Morning.
Good morning and welcome.
Thank you.
Sir, with your kind permission, we can start now.
So this is live on YouTube also, na?
Yeah, this is live on YouTube also, sir.
Okay. And
Sir, yes, sir.
Karthik, I have requested every staff of our college, senior college and junior college, including CHBs, to join today's seminar, national seminar, compulsorily. So we will note down their attendance.
Sure, sir.
We will note down the attendance, how many, in spite of my notice, how many of them have attending this today's national seminar.
Yes,
because this is not only related to botany, but it is related, it is concerned to every human being on the earth.
Sure, sir.
So it is important, irrespective of any faculty, irrespective of any subject. This seminar, I feel it is important for everybody. That's why I appeal all these faculty members, whether it's a senior college, junior college, or the contractual lecturers or CHBs, to join this national seminar. And our eminent speaker is there, who is going to give us a valuable information about our life, about that will save our life, and we can save others' life also, right?
Sure,
So
sir.
you note down the attendance also. Let me know after this seminar.
Okay,
We
sir.
can
Sure,
start
sir.
now.
Okay, sir. Aparna, it's over to you.
Thank you, sir. Good morning to one and all. I welcome you all to this one-day online national seminar on biofertilizers, a potential approach for sustainable agriculture development. This seminar is an initiative taken by Internal Quality Assurance Cell and Postgraduate Department of Botany, J.M. Patel Arts, Commerce, Science College, Bhandara. JM Patel College offers UG and PG courses in Arts, Commerce, and Science in Bhandara, Maharashtra. The college is awarded CPE status Phase 2 by UGC, recognized Center for Higher Learning and Research, and awarded Star College Scheme, Department of Biotechnology, Government of India. Today, we are honored to have with us Chief Guest and Resource Person, Honorable Professor Siva Prasad Adhikari, former Vice Chancellor, Fakir Mohan University, Balasore, Odisha. Chairperson, Honorable Dr. Vikas Dhomne, Principal, JM Patel Arts, Commerce and Science College, Bhandara. I also welcome Dr. Karthik Panikar, Dean Administration and IQAC Coordinator of the College, Head of the Departments, Faculties, Researchers, and my dear students. I welcome you all once again. Now I invite Dr. Saida Parveen Qureshi, Organizing Secretary, to give concept note about this national seminar.
Ask कुरैशी मैडम to unmute. कुरैशी मैडम आप अनम्यूट कीजिए.
मैडम आपका ऑडियो बंद था. हाँ, चालू करिए प्लीज. अब इट्स ओके नाउ.
Respected principal of our college, Dr. Vikas Dhomne sir, our esteemed resource person, Honorable Dr. Shiva Prasad Adhikari, delegates, researchers, and students. A very good morning to you all.
Again it is unmute. Again it is mute. Unmute कीजिए आप. हाँ, okay, it's okay now.
National seminar on
It's okay, continue.
National Seminar on Biofertilizer, a potential approach for sustainable agricultural development, being organized by the Postgraduate Department of Botany with the support of the IQAC of our college. At the very beginning, I would like to thank the principal of our college, Dr. Vikas Dhomne sir, for his immediate approval to organize this online national seminar. We are grateful to Honorable Professor Shiva Prasad Adhikari for accepting our invitation and consenting to be the resource persons of the seminar. Thank you, sir. Dear friend, as you all are aware, to meet the demands of the ever-growing human population, we are using synthetic fertilizer extensively. Various studies have conclusively proved that the long-term extensive use of this chemical fertilizer is not safe for human health. Dependence on chemical fertilizer for future agricultural growth would mean further losses in soil quality and possibilities of water contamination. Therefore, it is essential to use safe methods to increase agricultural productivity. In this context, one feels that biofertilizers are the need of the hour. The term biofertilizer refers to a formulation containing live microbes, which helps in enhancing the soil fertility by fixing atmospheric nitrogen, solubilization of phosphorus and other nutrients for plant growth by producing growth hormones. At present time, there is a growing concern about environmental hazards and threats to sustainable agriculture. A small state like Sikkim became the first Indian state to ban the import of all synthetic fertilizer, pesticides, and herbicides in 2003. A full ban on synthetic inputs went into effect in 2014 with Sikkim becoming India's first 100% organic state in January 2016. This ban has had a great effect on the immunity of the people of the state and massive reduction in the cancer cases. Sikkim today can be model for other Indian states and also to different countries in the world who want to go organic. Many well-meaning farmers and young professionals have in the last few years have switched over to organic farming. But extensive research is required to identify more suitable strains, develop better production technologies and quality control measures for wide commercializations of biofertilizer-based farming. The aim of the seminar is to discuss the important role and application of biofertilizer in the Indian agriculture sector. Biofertilizer are eco-friendly and cost-effective than chemical fertilizer. I am sure that we all will feel enriched with the valuable guidance of Dr. Shiva Prasad Adhikari and contribute in our own ways to make people aware about the need to change our cultivation methods. I welcome you all once again to the seminar and hope that you all will be benefited by this humble endeavor of ours. Thank you all.
Thank you, madam. Now I request Honorable Principal of the College, Dr. Vikas Dhomne, for the address.
Thank you, Aparna, and thank you, Qureshi Madam, also for giving us, apprising us about the threat of chemical fertilizers. Today's eminent person and resource person for this national seminar, Honorable Professor Shiva Prasad Adhikari, former Vice Chancellor of Fakir Mohan University, Balasora, Odisha. I welcome you here, sir. The organizing secretary, Kurishi Madam, of Botany Department. The head of the Department of Botany, Dr. Padmavati Rao. The organizing committee, the IQC coordinator, Dr. Karthik Panikar. The entire organizing committee, all heads of the department of J.M. Patel College, the faculty members of J.M. Patel College, the other colleges, those who have joined this important national seminar. I welcome you all. The couple of days ago, it's just 3-4 days ago, when Qureshi Madam and the head of the department, Rao Madam, approached me with this concept, with this idea about organizing a national seminar on the use of biofertilizer. Immediately I accepted it because I personally concerned about the fertilizers which have been extensively used in India. And out of my experience, when I travel abroad in so many countries, and when I compare with the Indian agriculture, so I get astonished, surprised about the vegetable and fruits which the European countries or America or Canada they are using as compared to Indians. We are far lagging behind it. India is basically known to be an agriculture country, as we always say that 80% farmers are there and having a sufficient agriculture and fertilized land. And we have a majority of farmers in India producing huge amount of food grains and vegetables and fruits and all. When the time comes now, this is the time when the world has started thinking about the global warming. The scientists have started thinking about the biodiversity, and then the idea comes of the biofertilizers. The biofertilizers are, at least for India, biofertilizers are not new for us. Since hundreds years ago, the Indian farmers are using cow dung as a biofertilizers in their fields for producing food grains, vegetable and fruits. And yesterday just I was searching the history of biofertilizers. So the history says that it has started around in 1895, long back. The biofertilizers have invented long back in 1895. The history says with the launch of nitrogen by Nobby and Hittner, a laboratory culture of rhizobia, followed by discovery of azobacter, and then the blue-green algae and the other microorganisms. Although I am not a student of botany, but Professor Adhikari will may understand what I am saying. Nowadays, our farmers are using extensively the chemical fertilizers. And there is a reason also. There is a reason of using chemical fertilizer, because the chemical fertilizers are cost effective. There are a lot of industries in India, those who are producing the chemical fertilizers. It is very easy to use. It is readily available in the market. So farmers can easily buy it at low cost. And then they just have to spread it, use it in their field, and the yield is very high. They are getting more production with chemical fertilizers. The more production of chemical fertilizers means they are getting more money. So just for getting more money, there is an extensive use of chemical fertilizer all over the India. But now the people have realized it has started in the European Union. The discussion has started long back. In fact, to my knowledge, most of the European countries, including the countries and USA and Canada, they have banned the fruits and vegetables coming from India. When I had been to Europe to visited so many countries, and I stayed in Canada for 2 months a year before, and then when I visited the vegetable market or the fruit market, so I was just asking them from where these fruits and vegetables are coming. In fact, I went there during the mango season in Canada, year before last year, and I was searching the Indian mangoes in Canadian fruit market. I was surprised to know that we are proudly saying that the Ratnagiri, Devgiri, Alfonso mangoes are famous all over the world. We are exporting all over the world, but I didn't get that Alfonso mangoes in Canadian market. The mangoes were there. Then I asked from where these mangoes have come. They said that we are importing mangoes from Mexico, from Brazil. They are importing mangoes and not from India. In fact, when I was traveling from India to Europe, India to Italy, and then Italy to Amsterdam 2 years back, I was carrying Alfonso mangoes for my niece who is staying in Netherlands. So I was asked not to at the Italy airport, Milano airport in Italy, during the check-in time, during the scrutiny, they have spotted that mangoes are there, and they said that you cannot carry the mangoes in your cabin. Indian mangoes are not allowed to carry in cabin. Then I put it in luggage and somehow I carry that mangoes. The reason behind, then I started thinking over this. What is the reason for this? They said that in India, there is an extensive use of fertilizer sprays and chemicals, chemical fertilizers and pesticides. And that is going to harm the human health. The research have proved it now. Research have proved that the vegetables, the food grains which are producing in India, the reason already I explained, because of the low cost readily available in the market and the high productivity, the farmers are using the chemical fertilizers. And that's why most of the European countries, the American country, the USA have banned the fruits and vegetable grains coming from India. It is now the time, because because when we think of our ecosystem and the global warming, the biodiversity is there, and to protect our earth, it is necessary to think of the biodiversity also, and then it comes to the biofertilizers. The use of biofertilizer, when we say it's a chemical fertilizer, then the soil quality gets degraded. Long-term effect. The long-term effect is the soil quality gets degraded because of the repetitive same crop production. The farmers are not thinking that the soil is degraded. Very few farmers are aware of the fact that there is a soil testing lab in every district. Government of India has provided a soil testing lab. Even in Bhandara, just adjacent to our college, there is a government department who is testing the soil for a nominal cost. But I'm sure very few farmers are approaching and getting tested the soil of their field. So this awareness is to be created. Although there are a lot of advantage and disadvantage of using the biofertilizers also. But we must think from the positive angle. We must see from the positive side of using the biofertilizers. As I told you, the cow dung, the vermicompost, these fertilizers, it seems to be a biofertilizer are extensively used in India since 1,000 years ago. But now, if you see today's newspaper, Lokmat newspaper, there is a big article that the for the fertilizer which is made up of cow dung, the cost is rise like anything. Today's newspaper here in Bandara, the Nagpur edition, Lokmat newspaper, Marathi newspaper, I read in the today's newspaper that the जो मराठी में हिंदी में हम शेन खत बोलते हैं, गोबर का जो खाद होता है, उसके जो प्राइजेस है, it has been increased like anything. Today's newspaper says. It means the awareness have started coming with the farmers that to use this biofertilizer and the cow dungs. There are lot many biofertilizers, but the thing is that the advantage is the chemical fertilizers, they are affecting our health. It is really a threat to our health. As in the theme of the seminar itself says that it may cause the cancers. The cancer patients have started increasing. We don't know, there are so many diseases that may cause to us because of using the vegetables and the fruits which are available in the market. I have observed 25 years back, I remember the size of apple, I remember the size of orange, I remember the size of vegetables 25 years back, and now today you see how the size has grown like anything. It is not the magic. Farmers have not done any magic, but they have used the chemical fertilizer. They have given the booster dose to the vegetables and food grains, and for getting good money, and ultimately that is harming our health, that is harmful our health. There are in Western countries, there is a strict laws. They cannot sell, the shopkeepers or the supermarket cannot sell any of the vegetable or any of the fruits. Everything get tested in the laboratory, and then the American government, US government allows. Even the toys and the equipments, the China is producing the highest equipments and the toys and the gadgets. But you see the difference, the gadgets which are exported to India and the gadgets which are exported to Europe and America by China, they are totally different. They are totally different because the quality of plastic which they are using for exporting it to European countries and the America, that is totally different because they have tested the quality, whether it is a toy, whether it is an electronic gadget or anything else which is manufactured in China, which is coming to India, it is totally different from the Western countries. So they are much aware that plastic may hazard our health. That is also a threat to our health and the global warming also. And that's why one should be cautious enough. Today's topic is very important, not for botany department or for researchers in botany and professor in botany, but it is in general. It is the health issue of in general, that which food we should consume. Now the concept of organic farming is coming up like anything. The organic foods are available in Indian market, whether it is a vegetable or the grains or the food grains. It is written there on the wrapper, this is the organic food or organic food grain or the vegetables. And people are giving the choice of purchasing it, although the cost is little bit high. The cost of organic food is little bit high, but those who are aware of this, those who know the advantage of this organic food grains or the vegetable, they are purchasing it, and now it has been getting popularized in India. So there is a time that the students, researchers, and the scientists like Professor Adhikari sir, it is now the time that we should create awareness among the students, because the students are the best messengers. They are the best messengers. If you literate our student, if you guide our students in a proper way about the use of biofertilizer and the disadvantage of this chemical fertilizer, so this message can be propagated. in villages, and the farmers can start using these biofertilizers. I remember a couple of months ago, there was a news in TV channels, very important, very interesting to share with you. As we say, China is one of the superpower in the world, in all the field. China is superpower, we are saying, but you know, the China's food production is very low. China is concentrating on the gadgets, electronics, atoms, and all except food grains. China has the highest population, and China is totally dependent on the food grains which has been imported from Australia and Canada. And because of corona pandemic, there was a conflict between Australia and China, and Australia has issued a threat to China that we are not going to supply you food grain, and China was having only the stock of 3 months food grain in their country. This was the news 3 months back. The entire China was having the food grain stock of only 3 months, and there was a threat from Australia that because of the corona pandemic, they are not going to supply these food grains to China. So the entire China gets disturbed, and then there was a mutual settlement, and they have resolved the issue. That is different story. What I mean to say, even the most powerful country in the world, China is the second most powerful country in the world, they are also dependent on the food grains. They are importing it from Australia, because these advanced countries like Australia, Germany is known for the manufacturing, agriculture equipment with very, very high technology. I salute to the German scientists. The equipments, particularly agriculture equipment, they are #1 in the world. The European countries or Canada or America or Australia, they are producing the food grain at the mass level, very high technology farming, they are doing it. And the workers are Indians. The workers are Indian in Australia, or whether in Canada, or whether in USA. The Indian farmers, the son of Indian farmers, they are working in their field. They are producing the huge grains, vegetables, and then they are exporting it from their countries, and ultimately their country gets benefited. So why India cannot do this? We said that we are the highest youth population, but unfortunately every youth wants government job. Friends, it is not possible that everybody will get the government job. One can focus on the agriculture product. One can focus on the agriculture issues also, because this is agriculture industry is one of the biggest industry, and maybe the most profitable industry. It will be profitable industry provided we should adopt the high technology, and it is possible only when we spread the messages, when we train our students, our scientists, and everybody, every stakeholder should note this and create the awareness amongst all, and then we say we can produce the crops or the food grains or the vegetable with the use of these biofertilizers. So today's session, I think it will be really interesting one, and I'll be happy to listen to our resource person, Honorable Shiva Prasad Adhikari sir. I'm really thankful to you for sparing your valuable time for JM Patel College, and for the other delegates, those who have joined from all over the state, from all over India. I have been told that there are so many delegates, those who have joined. So your message, whatever you will deliver, will be definitely helpful to us to save our life and to save others' life. Thank you very much, and thank you for inviting me, for giving this keynote address and listening to me. So whatever, although botany is not my field, but it is a general topic. That's why I could able to say something about this biofertilizer, biodiversity, the ecosystem, and the global warming. Thank you. Thank you very much. Over to you, Aparna.
Thank you, sir. Such an enlightening speech and your continuous support help us to take new initiative and guide us as a roadmap for this future. Thank you once again, sir. There is some instructions for the participants. All participants are informed to write their name and college name in the YouTube chat box. Participants are requested to put the questions in the YouTube chat box. Feedback link will be shared on the Telegram group. E-certificate will be issued to all the registered participants only after submission of feedback form. Now it's my privilege to introduce today's resource person, Honorable Professor Siba Prasad Adhikari, former Vice Chancellor, Fakir Mohan University, Balasore, Odisha. He was awarded gold medal in post-graduation and did PhD in botany from Berhampur University and also awarded DSc from Utkal University in 2000. He worked as a professor 2002 to 2009 at Utkal University, Bhubaneswar. He occupied various positions as a head Department of Biotechnology in Utkal University, as a professor at Vishwa Bharati Central University, Shantiniketan, West Bengal. His principal research interest in blue-green algae as a biofertilizer and marine algae as biostimulants for various crops. Both technologies' extension work is for the benefit of farmers. A lab-to-land facility established in village Maniakati under Surada block of Ganjam district since 1999. 27 students awarded PhD under his guidance. He published 165 research papers and 61 chapters in different edited books and authored 5 books. He visited several countries as a visiting scientist. He was awarded Samantha Chandrasekhar Award in Life Sciences by Orissa Vigyan Academic Government of Odisha in 1999. UGC Research Award again in 1999. Professor Harihar Patnaik Memorial Award in Environmental Sciences by Orissa Botanical Society in 2012. Dr. B.P. Pal National Environmental Fellowship Award on Biodiversity for by Ministry of Environmental, Forest and Climate Change, Government of India, and there are many more to follow. He is the president of many eminent organizations, and there are many feathers in his cap. It is very difficult to list them all here, but we are honored to have such an eminent personality today. Now I invite Honorable Professor Siba Prasad Adhikari sir to deliver the presentation.
Thank you, Madam Aparna.
Thank you, sir.
Respected Principal, Professor Dhomnik, IQAC Coordinator, Dr. Panikar, Madam Tulasi, who continuously interacted with me for this, the course coordinator, Dr. Padmanabhati Rao, Head of the Department of Botany, dear colleagues from the college and participants. I'm impressed with the profile of the college because it is one of the leading colleges of RTM Nagpur University. One of my friend, Balu Chopre, was the Vice Chancellor, so I know it very well. Secondly, RTM Nagpur University has a DBT HRD Center, and I was also the coordinator of DBT HRD Center for 10 years at Utkal as well as Shantiniketan. I had around 120 affiliated colleges when I was the Vice Chancellor. Very few colleges has this profile as yours, like College for Potential for Excellence by UGC. Also, a Star College Scheme of DBT. It is our feather to the cap to the college. I'm extremely happy for being invited. When I speak about this, I will use PowerPoint. But I will speak little different things. I will speak little science behind biofertilizers also, because it is national seminar. So I will speak on biofertilizers. I will speak little science behind biofertilizers. So those who are not from science faculty, please bear with me for 10 minutes, what I will speak about science. But I will confine on entrepreneurship. How you can start this program is a skill program in your college, so that you can come into the NEP 2020 for this. I will also a little bit about organic farming, about how it is linked to natural resources and other things while I will go on. So I will share the screen and the lecture. Let me open the PowerPoint, then I will share the screen.
Sir, is there any problem in sharing the screen?
Yes, so somebody has come up in between.
I have enabled the screen sharing option, so there shouldn't be any issue, I suppose.
Yeah, I don't think, I think I will just look at it.
Yeah, yes, sir.
You couldn't see anything, no?
No, no, sir.
Oh, it is not coming. I will do one thing. I will close, then I will start again. Okay.
Yeah, fine, sir. No issues. I mean, as far as we are concerned, it is all clear from our end.
Yeah. Okay. I will just look at it. Is it okay?
Yes, sir. Right. Yeah, it's okay. If you can
So
make it full slideshow, that would
I
be.
will just make it flat.
Yeah, yeah.
Is it clear?
Yeah, it's very clear, sir.
Fine. So the topic is suggested is biofertilizers, a potential for sustainable agriculture development. So actually, if you go for definition, the materials of biological origin commonly used to maintain and improve soil fertility are two types, two categories. I mean, any material of biological origin used to maintain fertility are two types. One is green manures, second is biofertilizers. Green manures are compost, vermicomposting, sesbania, all those things which are dead. But biofertilizers are always microorganisms, and these are living. This is the basic difference. All those things of biological origin, when we cannot call them as biofertilizers. These are all manures, green manures, organic manures you can call. But biofertilizers means these are specifically microorganisms. If I define biofertilizers in a stepwise way, say these are preparations of life, that means living microorganisms, or latent cells or dormant cells of what? Of efficient strains of either nitrogen fixing microorganisms or phosphate solubilizing organisms or cellulolytic microorganisms. These are used for application either on the seed or to the soil or to the composting areas. The objective is to increase the number of the organisms. That means you introduce live or latent cells and allow them to increase the number of these organisms. So when this number of organisms will increase, either on the soil or seed or composting areas, the metabolic activity will start, and they will make the nutrient available in a form which can be easily assimilated by plants. So this is a broad definition. What are the microorganisms? Where they are used? Why you use them? And they finally increase the fertility of these soils, and because plants can use them for their growth and development. So there are different types of biofertilizers. Mostly there are three categories. One is nitrogen fixing biofertilizers. That means that our atmosphere contains 70% of the nitrogen gas. But this nitrogen gas is inert. We inhale, but we cannot take. Plant can inhale, but cannot take. Okay, but only certain microorganisms present in the soil can trap this nitrogen gas and convert to ammonia. And these are again two types of nitrogen fixing biofertilizers. One is rhizobium. They always grow as a nodule in the legumes. And second one is azotobacter, azospirillum, BGA or blue-green algae, what you call as cyanobacteria or azolla. They're growing in free-living forms on the water or on soil, and they're only for cereals like rice, maize, etc. So these organisms are free-living, grows on soil or water, and these organisms grow in symbiotic association with the leguminous plants as a nodule. Suppose you uproot a mung or a mung type of plant, you will find some nodules in the root. Inside that root, you get rhizobium. And these organisms fix atmospheric nitrogen and supply to the plant. Second group of biofertilizers are phosphate mobilizing biofertilizer. These are again two types. One is phosphate solubilizers. That means in the soil we have phosphorus, but these are in unavailable form. Plants cannot take it. These microorganisms convert this unusable form of phosphate to phosphorus, which plants can take. These are called as phosphate solubilizing bacteria. Another type is phosphate absorbers. For example, in forest areas or in plants, big plants, the root do have a lot of fungus attached. This fungus grow in the soil, absorb phosphorus and give to the plant. These are phosphate absorbers or mycorrhizae. Your RTM Nagpur University group, they are specialized in mycorrhizae. And another scientist is in Anantapur, what is that called? Amaravati University. It's Mahendra Roy. He is a little expert in these PSBs. And third type are cellulolytic or organic matter decomposers. That means any plant dies, they have to be decomposed, get composed. So these cellulomonas or trichoderma, they decompose the plant cell, and these are cellulolytic microorganisms which produce compost. And lignolytic, these are a very rigid wall of some plants. This cannot be degraded easily, whatever method you may follow. But some bacteria, like Arthobacter, can degrade this lignin and make it available as a compost. That means biofertilizers are not one type. There are three types: nitrogen fixing, phosphate mobilizing, or making organic matter, or making compost. These are also some bacteria. So these are three types. They have all different properties. All bacteria do not have same properties. For example, rhizobium, which forms a nodule in the legumes, they fix 50 to 200 kg nitrogen per hectare per year. For good crop, we need 60 kg of nitrogen. But rhizobium has the capacity to fix 50 to 200 kg depending on the environmental condition and crop. And it increases the yield of the crop by 20 to 35%. Most important one, after the crop is over, they leave 40 to 80 kg of nitrogen to the field for subsequent crop. That's why from time immemorial in India and most of the agricultural countries, they make a crop rotation. First, they grow cereal like rice, and after rice, they grow legumes. So that legumes do have rhizobium, they fix nitrogen, increase the productivity, and leave 40 to 80 kg nitrogen in the soil left over. So subsequently, when you grow rice, you will get enough nitrogen to grow.
कॉलेजमध्ये खूप वेळा विचारपूस करतो कॉलेजवाल्यांनी म्हटलं की डाउनलोडच नाही होत आहे.
Please ask to unmute. Somebody is talking. Please, somebody is talking.
वीना महाजन मैडम, आपका माइक्रोफोन चालू है.
Sorry sir, sorry sir.
So second is BZA or cyanobacteria. They always grow in water, and they add up to 20 to 20 kg of nitrogen per hectare in the rice field. That means BZA you can grow, you can apply as a biofertilizer only for rice because they grow only when there is water. And rhizobium you can grow or apply only to legumes because they grow when there is no water. And BZA, Azotobacter, Azospirillum, these are all free-living microorganisms. They fix nitrogen as well as they secrete growth regulators like indole acetic acid, indole butyric acid, gibberellic acid, and vitamins, etc. to the soil, so that this water, soil into water, from that plants can take and grow. Azolla is a fern, water fern. It grows also in plenty when water is available. And this azolla has an endosymbiont inside the body, a blue-green alga, which fixes nitrogen. But azolla has a typical capacity, decomposes very fast. So you grow azolla, take to the azolla to the embankments, keep it with just 1 day. They will decompose fully and release a lot of nitrogen, phosphorus, potash, and increase the productivity of the soils. That means each biofertilizer, they have a different type or different environment to grow. Their capacity is different. They do different type of jobs. Here I will tell little science. Those who are not from science background, please bear with me because seminar I have to speak little science too. This nitrogen fixation, I mean what fertilizer you get, these are all urea. When you see the urea packet, it is written 60N. That means in a packet of urea, you get 60% nitrogen, rest 40% are carrier, like straw, like sawdust, etc., or like even sand. So this urea is produced in industries, chemically industries. But nitrogen fixation process means when molecular nitrogen or nitrogen from the atmosphere, nitrogen gas, is converted to ammonia. And here they need energy. To convert 1 mole of nitrogen to ammonia, they need 91 kilojoule per mole nitrogen. So much of energy is required. And this process is done in the industry, fertilizer industry. The process is called Haber-Bosch process. In this process, in the industry, when you keep 200 bar pressure at 500 degrees Celsius, we give a catalyst, nitrogen gas from the atmosphere will be dragged to the industry and convert to ammonia. That means in a fertilizer industry, there is no raw material. Raw material is the nitrogen gas or air. When you give high amount of pressure and energy and temperature, this nitrogen from the air will be dragged and convert to ammonia. This is the industrial process. There is no raw material required in this. So scientifically, this conversion of nitrogen to ammonia is a 4-step process. And biologically, when it's done inside a microbial cell, they need huge amount of energy, like 8 protons, to convert 1 mole of nitrogen to ammonia. In this, hydrogen is evolved. Hydrogen is a potential energy which can be also used as energy to run vehicles. Hydrogen energy is produced in many places. In many advanced countries, they do have hydrogen fuel to run vehicles and to even cooking gas. But the problem is hydrogen is highly dangerous. You cannot contain it. This is a different story and different science. I will not speak anything about that. So here you need so much of energy to convert 1 mole of nitrogen, and the enzyme, because in biological systems, everything is done at a controlled temperature and pressure. In industry, when they require 500 degrees Celsius, in a microbial cell, it is done at 25 degrees Celsius. And in industry, when they need huge amount of pressure, here there is no pressure at all. It's an enzymatic process. And this process is being carried out by an enzyme called nitrogenase enzyme. This nitrogenase enzyme is a very, very complex enzyme. It has 2 units. One is iron protein nitrogen reductase enzyme. One is iron molybdenum protein nitrogenase enzyme. When both the proteins are together, then only this nitrogenase enzyme is functional, otherwise not. This rhizobium occurs in the leguminous plants on the roots as a nodule. When these nodules are healthy, you can tell this plant is good and fixes more nitrogen. And these nodules are also found in stem in some plant like Sesbania rostata. This stem produces nodules, and inside it there is rhizobium which fixes nitrogen. Here in Bradyrhizobium japonicum, here inside these nodules, you have this bacterium which fixes nitrogen. That means there are either stem nodules or root nodules. These rhizobium are always growing in soil. In all soils, rhizobiums are present. But when you use huge amount of urea or DAP, the rhizobium vanishes. It is getting destroyed. It's not usable. That's why when there is no rhizobium in a fertilizer enriched soil, we supplement nitrogen to the soil. So we supplement rhizobium with soil as a biofertilizer. The story goes like this. In 1964, there was a novel by a Hungarian novelist, not a scientist. He wrote a novel in 1964 predicting unless you produce more by turn of the century, half of the population will starve or will be out from hunger or suffer from hunger. So looking at this prediction, many people got panic and started working on biofertilizer. At that time, Green Revolution started in India. Swaminathan did it with high-yielding varieties, but high-yielding varieties won't produce more unless you give fertilizer. At that time, our requirement was to produce more at any cost because India was starving. So obviously, fertilizers were used. When too much of fertilizers are used, almost all beneficial microorganisms present in the soil since several centuries, they all perished. And by 72, when analysis was done, our soils were already degraded. You know that June 5th is celebrated as World Environment Day. In Stockholm, in 71, World Environment Conference was held. There it was predicted that because of high use of fertilizers, most of the soil gets degraded. That's why the biofertilizer concept came. What is biofertilizer then? We have to go to the soil, find out whatever rhizobium present or living still, get those things in culture, check whether they grow fast and they fix more nitrogen, make a package, and again introduce to the soil, or again introduce to the field, so that they will do their job of nitrogen fixation and you will get more productivity. This rhizobium is a gram-negative bacteria. It is obligate aerobic, chemo-organo-heterotroph or chemo-litho-autotroph. And this growth period in the soil is very, very slow. That means this bacterium grows in soil doubling time. One cell to become double, it needs 10 to 15 days. But once it enters the plant to form a nodule, growth is very fast. It doubles at 9 to 10 hours only and forms a nodule. Very interesting science happens. The plant root hair recognizes rhizobial cell present in the soil. So when a rhizobial cell finds a root hair, there is a communication goes on between them. So this bacteria secretes a nod factor by the bacterium, so that when the nod factor will reach the plant, this root hair will curl like this. Once the root hair will curl, the bacteria will go and sit on this root hair. And then this will go inside the plant as a thread and finally form this type of nodules. Once these nodules are formed, means bacteria are present here and they fix atmospheric nitrogen and supply to the crop plant. And these nodules do have a typical structure called bacterioid. Here, a anaerobic condition is maintained. There is no oxygen because nitrogenase enzyme is very, very sensitive to oxygen. This inside the nodule, inside the bacterioid, such condition is maintained so that there is no oxygen can enter. That's why here nitrogenase enzyme is active and fix atmospheric nitrogen. I will skip a little bit. These are total science because I know that the audience belongs to several fields. So what happens? The bacteria gives a signal like polysaccharides, and plants gives a signal like lectin when they come together in the soil. Then plant will produce flavonoids, and these flavonoids then will induce a nod gene. This nod gene will build a nod factor in the bacteria, and this nod factor is mostly a carbohydrate complex, and this nod factor will develop this nodulin protein and nodule is formed. Then only nitrogen fixation will occur. And to activate this process, that in energy they get through photosynthesis. I will skip this one. So first there will be a reduction of the protein, then the iron protein complex will build, then the enzyme will be formed and reduced, and nitrogen fixation ensures, and along with that hydrogen is produced. So this is the entire nitrogen fixation process. The nod gene is a very, very complex gene. It is composed of 16 genes. Pure information to all of you. So by 70s, when we were young, when we were young students, the best science was at the time how to transfer a NIF gene to higher plant. Because NIF gene, which fixes nitrogen, is present only in bacteria. If you transfer that gene to a rice plant or a maize plant, plant will fix nitrogen. No need to give fertilizer. So how to transfer this gene to from the bacteria to the plant was the leading science. Like now, the leading science is to develop a vaccine for COVID. Few days ago, the best science is how to develop CRISPR-Cas technique to have high productivity. Before that, to sequence the whole genome of human being, human cells or human chromosome to be sequenced. Like that, every time there was a science. During 70s, when we were a student, the best science was how to transfer the NIF gene from the bacteria to the higher plant so that fertilizer is not required. But the problem is, if you transfer a gene from a bacteria to a higher plant, you need a vector, you need a virus. The vector can carry only 4 genes. But when the nod gene is 16 genes, how can you transfer? That's why the entire science failed. Even the NIF gene is not a small gene. It is a complex gene, 64 genes. And each gene has one factor, one function to do. And how to transfer a complex of a 64 gene to a plant from a bacterium? That's why it didn't fail. So entire science for 10 years failed, but we could know several informations about this biofertilizers, how to do it. This is blue-green algae. This grows in rice field. You find that there is a specialized cell within the cells. This is the specialized cell, this one. And in this specialized cell only, nitrogen fixation occurs. That means in this specialized cell or heterocyst, there is no oxygen evolution because there is no photosystem 2. That's why nitrogenase enzyme is active. Here it fixes nitrogen. So in this blue-green algae, nitrogen is fixed in this cell, transported to the neighboring cell, stick it out. And these cells are quite different. And heterocysts do not have photosystem 2. It was developed by Indian scientists. That too from Bhabha Atomic Center, Joseph Thomas, published in Nature. And I will skip these things because these are all complex things. I will not speak. Okay, now let us come to the science. What do you all please ask others to mute because I'm hearing the voices. Please ask to mute, everyone to mute. So now let us come. This is for everyone. In a rice field, whenever you get little water, say 5 to 10 centimeters of water, you get organisms grow like this. These are patches. These are not green, but slimy patches. You will touch them, they will slip away from your hand. And these blue-green algae, when they grow in rice field, they fix atmospheric nitrogen and supply this nitrogen to the crop plant. You know, in India, this part, wherever I have put a black one, here rice is grown predominantly. And here the people eat twice rice, morning rice, evening rice, even the night rice. They only eat rice. Say some part of Assam, West Bengal, Bihar, Eastern UP, some part of Chhattisgarh, Jharkhand, Odisha, Andhra Pradesh, Tamil Nadu, Karnataka, and Kerala. And Maharashtra areas, you eat half a day rice, and after you eat wheat, you know. But we belong to this area where rice is very, very important. And here, rice is grown twice, sometimes once, and in water fields. That's why here, if you want to use any biofertilizer in this part of the world or in this part of India, only biofertilizer you will use is either blue-green algae or azolla, because they grow only when there is water. I will come to the other ones also for what is necessary for Maharashtra also. This biofertilizer work in India, blue-green algae biofertilizer, started as early as 1939. This P.K. De was a professor of Allahabad University. He found that in UP and Bihar, people grow rice year after year without using any fertilizer. Fertilizers were developed only after 64. Before that, there was no fertilizer. Chemicals were there, but fertilizers were not rampantly used. So this publication in Proceedings of Royal Society of London, P.K. they told that because of blue-green algae grows plenty in UP and Bihar soils, that's why they fix nitrogen and rice grows well. Then another scientist, Professor Ram Nagina Singh of Banaras Hindu University, professor of botany, he wrote a book, Role of Blue-green algae in Nitrogen Economy of Indian Agriculture. It was published by ICAR. And here he emphasized what important role BGA plays. Then at IRI, one scientist, G.S. Venkataraman, in 1972, started working on algal biofertilizer rice cultivation. He wrote a book. And the technology was developed by him and his group in 1978. It was algal biofertilizer for rice, a information bulletin for farmers. And this became so popular in India. He wrote another one a large volume about the algal biofertilizer of rice. It was a production manual. It was published by Food and Agriculture Organization in Rome, FAO Rome. So this information manual of 8 pages developed by Venkatraman in 78 at IRI looks like this. Here, this is the blue-green algae. These are the heterocysts where nitrogen is fixed, and this is the rice plant. This shows that these organisms fix atmospheric nitrogen, release nitrogen to the soil or water, and this nitrogen is taken up through rice roots and plant grows. This is for farmers. And the technology he developed is like this. This is for all of you. Now you go to any agriculture office anywhere in India, any district agriculture office, you get this type of cemented tanks. These are directly under open sun. These are 3 meter long, 1 meter wide, 10 inches deep. The technology is Venkatraman and his group in 70s, they went to all over India, collected whatever blue-green algae grows in the soil, then cultured them, and they got around thousands of blue-green algae from Indian soils, cultured them and checked which is the best nitrogen fixer and grows fast. Basing on that, they have selected 5 different blue-green algae and made a package. They told that you make this type of tanks, take 10 kilo of soil, put water, and mix the soil well. Then put little superphosphate, 250 milligram of superphosphate, so that this phosphorus is required for any organism's growth, even for blue-green algae. Then put little puradon granules so that mosquito larvae won't come, or mosquito won't come and give breed here. They will be killed. And after this, when the water will settle down overnight, then put these 5 blue-green algae they have selected for Indian conditions to use as biofertilizers. You put these 5 algae here and allow it to grow under sun. These are photosynthetic organisms. They will grow. Within 7 days, they will grow the entire pit. They will grow like this. Every day, some water will be evaporated. Put water so that 7 days you go on doing like this. And in 7 days, entire pit will be full with the blue-green algae patches, mixture of 5. Then don't add water, and they will all dry down. When they will be dried fully like this, you take it out and make packs. These are BGA biofertilizer for rice. Each one is 1 kilo. That means from 1 pit, you can get 10 kilos because you have taken 10 kilo of soil and blue-green algae and grew for 7 to 10 days. So each packet, 1 kilo, is sufficient for 0.2 acre of land. Or for 1 acre, you use 5 packets of biofertilizer. How to do it? You transplant rice, and after transplantation, when these plants will become firm and the field will have 5 to 6 centimeters of water, then you sprinkle this biofertilizer to the field. So they will grow, multiply in the field, fix nitrogen, and supply nitrogen to the crop as well as hormones and vitamins. This was the technology. So this technology developed in 78, then popularized. In 82, almost all district agriculture officers has BGA production units. But demand grew. When the demand went on, how so much of cemented tanks will be built? So then what they did, they put this polythene setting in a pit in the field and grew blue-green algae in these pits. Still demand grew. Then what they did, they make these fields level without any polythene. They grew the blue-green algae. In biotechnology or any technology, if quality of the organism is not okay, the technology will fail. Here, quality control was compromised because they didn't put any barrier. So whatever organisms present in the soil also grew along with the blue-green algae. So this is the problem why by 5-6 years, the entire program failed in India. But it became a bumper success in Tamil Nadu, Rajasthan, and Haryana. Because most of the organisms in that pack they given by IRI were strains from those areas. Because Venkatraman is from Tamil Nadu, BD Kaushik is from Rajasthan, and SK Goyal from Haryana. So they took most of the strains from those areas. They grew well in their ecological conditions. And in eastern part of India, the entire program failed. So this is story I'm telling of 87. In 87, I joined as a assistant professor or lecturer at the time at Utkal University Botany Department. At the time, I was already having a PhD in 80, and I was having a postdoc in Germany for 3 years and in Japan for 2 years. And after coming back from abroad, when I joined Utkal University, and I wanted to know how this biofertilizer program works, I went to an area nearby how they produce biofertilizer. It is very close to Bhubaneswar and Cuttack. It is written Neela Harit Saivala Utpadana Kendra. You understand what it means. So here they put the fields like this, and then whatever green things grew and dried once in the field, they packed and given to the farmers. I checked what organisms grow there in the laboratory. I found of the 5, hardly one is there or none. And we find mostly green algae diatoms are growing, and green algae and diatoms are nitrogen consumers, not fixers. So this the condition happened because quality control was not taken into consideration. Entire BGA biofertilizer program in Eastern India failed. Then why? Because quality control measures were not taken. Inoculated species in most situations failed to compete with the local species. An increase in grain yield due to cyanobacteria inoculum was not consistent and varied under agroclimatic regimes because all states has different agroclimatic regimes. Then I asked the question, how to ascertain that inoculum species successfully established in the field? And what should be carrier to be taken instead of soil? And how the regulatory authority will prescribe standards? With this question in mind, I started doing work with the support from DBT, Government of India. What we did, we got a project, R&D project from DBT. First, what we did, we surveyed almost all soils of Odisha, 10 agroclimatic zones of Odisha, what nitrogen fixing blue algae grows in the rice fields fast. Then isolated the organisms, checked their growth and nitrogen fixing ability in pure culture in the laboratory. They established a germplasm collection or cyanobacterial germplasm collection at Utkal University, Bhubaneswar. Then if you want to use a biofertilizer, we screened the best. When we want to screen the ecologically compatible and stress-resistant strains the best to use as a biofertilizer to be applied to the field, we checked those organisms grows both in Rabi as well as Kharif season. Those who grow better and have higher nitrogen fixing ability, and do have a wider tolerance to pH and temperature, because soils are not of same pH. Some are acidic, some are alkaline, some are neutral, like that. And capability to tolerate high salt. Odisha has 460 kilometers of coastline. In the coastline, these are all having a lot of salt. So those who have high concentration tolerate to salt. And they have the potential to secrete high amount of extracellular substances while growing. Fix nitrogen and secrete them out. Those have the capacity. And those who can grow mixotrophical. That means when you give organic carbon, like all villages nearby do have rich with carbon fields. And those with carbon and light they get, they grow very fast. Those organisms do have this capacity and tolerate high concentration of the pesticides and chemical fertilizers used by the farmers. Farmers use a lot of carbamates, organophosphates, organochlorine, DAP, SSP, ammonia, urea, so many. So those organisms do have little higher concentration, higher tolerance capacity to these pesticides and fertilizers. Odisha is rich with iron, Maharashtra too. Odisha has around 70% soils, 70% iron is present in the soil. So those organisms do have high tolerance to iron and quickly form spore and survive desiccation, like your Vidarbha area, a lot of dry areas. In those, you need these organisms which readily form spores or have desiccation tolerance. With so many characteristics, we went on doing research to find the best organisms. Few people got PhD on this. What it did, we went to all part of Odisha, wherever there is a dot, we went and collected soil samples, then cultured them. We checked which areas are rich with blue green algae, which are less with blue green algae, which are deficient with the blue green algae. And those areas which are deficient with the blue green algae, we concentrated that to be used as a biofertilizer. For example, this area already blue green algae is present in the field, not necessary. But here they're totally deficient, or here they are very less. There we wanted to use as a biofertilizer. First, we isolated them in the laboratory, identified, gave a strain number, U is Utkal University, check the doubling time at how many hours they double, that's the growth rate. Quicker means they double quickly, say 72, 82 hours like this. And this is nitrogenase activity, how much of nitrogen they fix using a gas chromatograph. We have managed everything to check all. And finally, we treat them in a cultural laboratory. All these things maintain the strains, even in immobilized state in sodium alginate beads for experiments. And then we selected finally 5 Bruggen algae from Odisha for use as biofertilizer. But here it is 7. Why I have told 5? Reason being, after you transplant and put this biofertilizer, first this organism will come, where there will be around 1 feet of water between August. This organism will come. This is cylindrosperm. After that, during, say, before panicle emergence, you get this organism. This is Anabina. Both are different Anabina species. At the time of Rakhi Purnima, when the panicles will already emerge, you get this nostoc. When the water will be dried up, ripen season, you will get Calothrix. And when the soil will totally dried up, right now, November, early December, before harvest, you get Alocera. So we got, this one is from coastal Orissa, this is from western part, very close to Chhattisgarh, very close to your Gondia, your area. And this is from Odisha, and this is from Odisha, this is from coastal zone, this is from western zone, very close to Chhattisgarh area, Sambalpur, all those areas. So we developed a package. That biofertilizer we use for coastal region, you take this, and this. And for western zones, where the dry area, we take this one, this one, this one, this one, and this one. So like this, we developed biofertilizer packages and developed the experiments. We gave the report to the DBT. It worked very well. We got 15% higher yield. Science is okay, published papers, had publications, even had PhDs. But how will we release the people? I told DBT that you give me a project where I will take the technology to the people. DBT first told that we won't give because you are a scientist working in a university. Why shall you go for people? Agriculture people will do it. I told agriculture people do not know. They never do this. And unless you give money, all the 1 crore rupee you have given me, everything is wasted. And finally, they gave me a project to work with the people. So I went to a people village in Ganjam district, in the remote area. You find it's a very remote area. And I reached there and started popularizing the organism. This you can do in colleges, in extension program. So in the village, what I do, I used to take these organisms in a small tube, then grow in conical flask. But to make it cheaper, we cultivate it in polythene bags under light. This is a small laboratory developed in a village, and we had semi-poly houses. Because why semi-poly houses? Because earlier technology of IRI, when we make it open, sometimes birds will come to take bath. Sometimes you will find these snails will climb to eat algae. So that's why you made a semi-poly house, so that air can pass easily and even the rainwater cannot enter here, and made this type of pits. And we never allowed all the 5 cyanobacteria to grow together. Suppose in this pit you greet one organism, here another, here different one. Why? Because if all these 4 are best, they will always compete among each other, overpower the other. Like if there are 4 students, very good students, staying in a hostel in the same room, what they will do? They will compete. One will try to overpower. And finally, one will become succeeded, and other 3 won't be of so much of potency. Same thing happens with science, with the biological organism. That's why we grew all organisms, all the 4, in different pits. Within 7 days, they grew very well and then dried. When they become dried or latent, they won't compete. And after they dried, we mix them and make a biofertilizer. Here we took not garden soil. When a garden soil already has some biofertilizers or bacteria, they may sometimes interfere. That's why we took brown, red soils, forest soils, where there are very little microorganisms. So those things we took and make these biofertilizer packages. And then you tried in the field. When you tried in the field, once after harvest, you find these organisms established in the field, your program is successful. Because of fertilizer use, all the microorganisms have been depleted. But when you introduce again, and you find after harvest, these organisms settle down in the field. Okay, this is one type of cyanobacteria, this is another type of cyanobacteria. Then your program is successful, and you need not put biofertilizer after 2-3 years. And we got 10 to 15, 12% of high yield only by using cyanobacteria biofertilizer. This we published in newspaper in Indian Express in '98 that a new type of biofertilizer developed by us, and we had 120 brunal strains from Odisha maintained in germplasm collection and supported by Department of Biotechnology, Government of India. And here 60 strains we have even deposited in IRI, New Delhi. And this is the packet. It has BGA biofertilizer, BGA Jaiva Khad. This is in Odia. Polyfer rice produced in this particular place and produced by Department of Biotechnology, Government of India. And these are in Odia for the farmers, how to use them in the field when they will use them in the field. Most important is training. When you go to a village for training, usually women never come to you because they have reservations. This is a picture of 1999. I was young at the time. And I tried to train people along with some of my co-workers. Even some of my scientists at my university, I took them to the village and told the ladies to explain the ladies. And even made a lot of training programs. This was a vice chancellor of Utkal University, was in the village. This was another vice chancellor. This was a principal of a college, very leading college, BJB, Bakshi Jagabandhu Vidyadhar College, very leading center for excellence. I wonderful college in Bhubaneswar. And this is a lady from Ramadevi University. All these persons went to different times in that village and to give training to the people. Even MLAs, MPs, scientists from CRRI, OU at the Agricultural University, they all came and trained in this village. Many of the students of different universities came to that particular site to get the training on biofertilizers in different times. Even once I got the NGOs of different parts of Odisha, around 30 NGOs, they came, stayed 3 days in that village, and they were trained how to produce biofertilizer and become entrepreneur, sell them, and get the benefit to become an entrepreneur. So we developed a self-employment rural sector technology using this. For example, unemployed youth, SSGs, NGOs can produce this biofertilizer for income generation. This can be used as a cottage industry scale in Gram Panchayat level for self-employment. We have calculated by investing 1 lakh rupee for infrastructure and working capital of around 25,000 per year, one can get net profit of 10,000 rupees per month. And why? Because 5 kg of soil-based biofertilizer or 1 kg of soil or crop-based biofertilizer with requisite number of viable blue green algal cell is sufficient for 1 acre of land soon after transplantation. How this is calculated? For example, if you make 25 tanks with brick and cement, the cost will be 60,000 rupees. Iron framework with polythene setting 30,000 rupees. One compound microscope is a mask 10,000 rupees. Total investment for infrastructure is 1 lakh rupee. Recurring expenses, you need superphosphate, lime, insecticide for 25 tanks per year, 3,000 rupees. Carrier material, soil, core, etc. if you buy 2,000 rupees. Polybags for packing 5,000 rupees. Labor cost, now Odisha, it is around 200 rupees. At that time, it was 150. You take labor, employ a labor for 100 days only while harvesting. So it is 15,000 rupees. Total investment of recurring expense in a year is 25,000 rupees. So if you calculate and if you sell 1 pack of biofertilizer at the rate of 25 rupees, so you can get 24 harvest, 15 days one harvest in a year, you can have 6,000 kilo of biofertilizer. If you sell 1 kg for 25 rupees, selling cost of 6,000 kg will be 1,50,000. So net profit will be 1.5 lakhs minus 60,000 infrastructure, and total profit will be 90,000, or say around 7,500 rupees per month. One can get net profit from 25 tanks per person if they use a biofertilizer. One can even go for a medium scale industry. You can have a laboratory requirement of 20 or 200 feet to 200 feet. In the laboratory, one room can be, in the college, one room can be devoted for this. Laboratory, very small one, 15 to 20 feet of laboratory, 300 square feet. One storage for carrier, store for biofertilizer, one office cum cell counter. Building cost say will be 15 lakhs. Furniture accessories 4 lakhs. Poly house, if you make it semi-poly house, 10 numbers, 10 lakhs. These are the equipments required, around 6 lakhs. And you can employ, these are the pre-operating cost. You can put 4 skilled workers, 1 production manager, 1 marketing staff, 1 watchman. Say salary will be 10,000 rupees. Technical know-how is free. Everything is available. And total budget will be around 50 lakhs. Net return will be around 1 lakh rupees per month. If a skill component can be added in your college, you can do it, or you can train entrepreneurs or unemployed youth. They can do, even 2-3 SSGs can come together and start this biofertilizer industry. I have calculated in Odisha state, we have 2,500 gram panchayats. If each gram panchayat will have 2 units, you can meet the biofertilizer demand for rice, plus overnight can employ 5,000 youth to become entrepreneur. So this is a technology. This is not like the people, SSG people taking money and giving loan and telling that we are SSGs doing this, that. If you train little matriculate persons, little know the science, technology can handle 2 units per panchayat, 5,000 units come overnight in an area and can produce biofertilizer and can meet the demand. This is about BGA. But other biofertilizers are also produced by IRI. These are the biofertilizer inoculants produced by IRI in 78, like rhizobium inoculant, azospirone inoculant, etc. How these are produced? This you can do it from 2005 onwards. We are doing this even for rhizobium, azotobacter, azospirone, etc. for rural demonstration. The same laboratory looks like this. We have a refrigerator, 1 incubator, 1 oven, 1 spectrophotometer, 1 sealing machine, 1 small fermenter, shaker, etc. And this can be easily handled in a laboratory. And you can even, using this laboratory looks like this. We take strains from Utkal University, grow them in this incubator and oven, oven you dry this thing, incubator you grow, or even the Saccharose glasses or in fermenter, and then make the technology. We make media, autoclave them, all in village. Okay, after autoclaving, we put it in incubator. In 3 days, you get enough cells. Then make a mixture of red soil and charcoal 1:3. This red soil and charcoal powder you mix with this bacterial culture, and then mix well, dry insert this way, and after this biofertilizer is dried, is azospirillum or azotobacter or rhizobium, all same technology. And then you make 200 grams pack each, and this 200-gram package you seal and sell it. So these are the different steps how to do it. The biofertilizer, the 200 grams pack of rhizobium or azotobacter or azospirillum, what you get, you use as a seed treatment, mix with seed and plant, or a seedling, grow them, tip the roots and plant, or even you can amend to this soil so that you can have the productivity. So production technology we have standardized. First, you take the bacteria culture in slants, kept in refrigerator, prepare the mother culture at 37 degrees Celsius for 5 days. You revive this and make a mother culture growth of little higher, 500 milliliter capacity in a rotor shaker, and inoculate this actively growing shaker to a glass fermenter of 10 liter size. Intermittently, the culture can be checked for purity, and the harvested bacteria mixed with the carrier with the lignite and charcoal at the rate of 1:3, and 1 liter of culture mixed with 5 kg of carrier and air dried, and make biofertilizer packs 200 grams each. The cost benefit we have calculated: one-time investment for infrastructure for fermenter, refrigerator, rotary shaker, autoclave, microscope and air conditioner is around 2 lakhs rupees. Recurring expenses here, return from 10 liter capacity of fermenter, one can have 50 kg carrier based biofertilizer or 250 packets, 200 grams each. In 4 harvests in a month, you can have 1,000 packs. Production in a year can be 12,000 packs. Selling cost of one will be 15 rupees. 12,000 packs, 1,80,000. Investment 30% use minus. So say you can get 10,000 rupees per month if 2 persons will work for this biofertilizer. I know many people in Nagpur area are producing biofertilizers, but these are not pure. These are companies, they never, maintain quality. India government has regional biofertilizer development centers, 5, of which one is in Bhubaneswar, one is in Nagpur, one is in Pune, one is in Manipur, and head office in UP, somewhere very close to Lucknow. Even then, the quality is not maintained. If the quality of biofertilizer is not maintained, you will just buy the carriers, not biofertilizers. But this is easy. If anybody little science know about the bacteria can use it. So the glass vessel, we have bought it by ourselves, and this was developed by Regional Center for Organic Farming at Nagpur, very close to your area. This is a low-cost ferment was produced. It's a Boracil glass vessel. Catalog is so of 10 to 20 liter capacity with a stainless steel head. Membrane filter is given, air inlet is given, one aerator we can put, and you can fabricate fermenters. This looks like this. I had a friend who was director of Regional Biofertilizer Development Center, Dr. Yadav. He showed me all these things, and he has a book on this. So you can follow your area, you can follow and produce this in the college itself. So as I told you, this biofertilizer can be used in soil, in seed dressing, or make seedlings. Take your biofertilizer in a bucket and dip, and then plant. Even for flower, even for other crops, you can have nice yield using biofertilizers. So in that village now, what do we do? We have a production unit for blue-green algae or cyanobacteria, and also inside the laboratory, Rhizobium, Azotobacter, Azospirillum. Always farmers look the result. Seeing is believing. That's why in that area we put this type of cemented tanks and grow these crops, various crops. This is a, groundnut using Rhizobium. Even you grow with Azotobacter, chili, tomato, brinjal, various types of biofertilizer are used, even rice, and tested almost all biofertilizers and showed the result to the farmers. Many times we ask people, village people to come, ask some agriculture officers from government to come and train them how to produce biofertilizer. And many times students come from different parts of the state, or our students go, interact with the villagers and see the facilities. In India, there is a typical problem. Indians never believe on Indian science. If you tell the science is from abroad, people think this is a very good science. I had taught in abroad several times. Around 11 times I went as a visiting scientist and as a professor, taught in Germany, Italy, Japan, many places. But in India, when you teach or when you tell a technology, people don't believe you. They always believe foreign technology. If estrogen will come from Oxford, people believe. If it will be developed in Pune, people won't believe, even after COVID. You see, this will come because our people will never believe us because of dishonesty. Many people's dishonesty. They think that everybody is dishonest. It's not true. So what I do, whenever I get a guest from abroad, I get a lot of guests abroad through collaborations. This is a lady from Rome. So when two ladies came from Rome, I always take them to the village. I tell them to speak what I want. If they speak what I want, people believe. So this is the typical. Many a times I get, this is a person from United States of America. I took them to the village and told them to speak. And because he do not know our language, our people do not know except Odia. So I will speak and he will speak, I will translate to Odia. Whatever speak, I don't mind, but I will speak whatever I want. And people believe that this is a tone of foreigner. And this is the person from Tata Energy Research Institute. These are the persons from Czech Republic. Many foreigners, whenever I get any guests, I take them and in different times and apprise the people. Finally, government of Odisha, to Rashtriya Krishi Vikas Yojana, they gave 2.5 lakh subsidy to 47 entrepreneurs. Odisha has 30 districts, but we have 47 agricultural districts. From each agricultural district, they identified one leading farmer, gave 2.5 lakhs rupees to produce BGA biofertilizer and Rhizobium. I took them from Bhubaneswar to that village and giving training right in the village. And this is the training right in Utkal University campus in the biotechnology department. Like this, we went on giving training to them. And this is another picture. These persons came from United Kingdom, from London. So I took them also to the village, tell them about the biofertilizers. And this from US, this is from Florence, Italy. You see, when they come, even they take pictures here and people believe this real science of ours. Many times whenever I go to the village, I ask farmers come and give training in the village, inside the room, and strains are distributed free. I given this strain to IRI, also Central Rice Research Institute, Cuttack, because that is an accredited institute that is known as National Rice Research Institute. Now they use these biofertilizers. Whenever we make a program, we always give training and advertise in newspapers. And this is a article in Odia. You cannot understand Odia, but I will read what is written here. It is BGA Joibosara O Biotechnology Silpa, or BGA biofertilizer and biotechnology industry. How to do it? Detailed technology is given by me, written my address there. So now Indian Bureau of Standard has come out. They have developed 28 standards for quality biofertilizer. For BGA, the IS is 14634 is to 1999. That in the pack must contain 10 to the power 7 cells per gram material, gram carrier. pH should be 6.5 to 7.5. The carrier should be soil or peat or lignite. The granule size should be 100 to 50 to 210 mess. Even one can take multani mitti as the carrier, or even straw can be taken as carrier. So there are a lot of research is required in this. We develop the technology, but still research is going on and required in many universities. We need competitive strains capable of establishing the rhizosphere. The strain must establish rhizosphere so that they will do their work well. The carrier material, what you take in the packet, should have high survival of the organisms. If the organisms won't survive in the carrier on storage, it will be a failure. Inoculation technique for delivering a high population of inoculant to the soil and rhizosphere is important, how to do it. And also you can develop a consortium of different cropping systems. Because if the cropping systems are different, agroclimatic zones are different, how you can develop the different consortia. So for carrier material, alternative carrier material, a lot of criteria has to be taken. The carrier should be non-toxic. They should have high adsorption capacity. They should have good buffering capacity. They should have to carry high microbial load. It should be cost effective and easily available. Microbial load of 10 to the power 7 sacks per gram carrier should be there. And these rice straw, wheat straw, coconut coir should be easily available. And multani mitti, if you get it, should be low cost, or you can take brown forest soil. Always, we need appropriate production methodology to follow strictly. This is very important for extension. Without extension, without the technology going to the farmers, technology won't work. Nowadays, what farmers do? They go to a fertilizer shop or pesticide shop. They will ask him what fertilizer I will use, what pesticide I will use. He is his master. What are the person selling will tell farmers use it because they have no idea about the extension. Quality of the inoculant is very important. Many industries produce biofertilizers which are not of quality. So once you don't get quality inoculant and produce in the field, they won't work. So obviously the faith will go. We need quality control laboratories. Always there has to be control. We need establishment of biofertilizer production units. So whenever anyone needs biofertilizer, they must be available. That should be adequately tested in the farmer's field. And most important, we need trained manpower. Our program, what seminar you are doing, this national seminar, just for this trained manpower. Unless the trained manpowers are there, you cannot develop any technology, especially agricultural technology. Farmers has a lot of concern because they lack knowledge, they lack awareness due to poor extension efforts. Biofertilizers are not available at the sowing time. When they need, these are not available. Many times they supply expiry packets. And many times the quality of the inoculum is very, very low. That's why farmers has concern on biofertilizer. Manufacturers also have a concern. Shelf life or storage life of biofertilizers is very, very short because these are living. Unless when you produce, if they're not sold, there is a risk of you get less profit. So there's a major concern of the manufacturers. If the carrier material is not attractive, people won't take. If you take charcoal, who will take? And because if you take charcoal and lignite, and farmers, the person who bake it, it gives a health hazard. The dust will go to the air, lungs. And carriers not using only natural resources, like multani mitti, charcoal, etc. These are natural resources. If you burn forest or get multani mitti, natural resources will be lost. That's why we need alternate carrier materials like polyethylene foam. Foams can be used, and this can be tried because these are not natural materials and they won't make any problem with the field. So, in general, if you see the quality, cell number is not only the criteria. Biofertilizer pack, what is available in market, should have levels. It should contain how much cells present, what it contains. So anyone can test and see whether your biofertilizer quality is good or not. It should be written to which crop it is used, because biofertilizer are various types. If you take rhizobium, put for rice, it won't work. If you take a BGN, do for legumes, it won't work. Okay, so biofertilizer, for what purpose it is required, whether it's for compost or for legume or for a cereal, it is written in the packet. And how much yield one can have upon application is to be mentioned. So if these are taken into consideration and quality is maintained, rigorous control is taken by government, then only there will be ultimate goal of acceptance by the farmers. Madam Qureshi, shall I continue or shall I stop? Please unmute. Please unmute. Shall I continue or shall I stop?
Yes, sir. Yes, sir. You can continue.
15 minutes more?
Yes, sir.
Okay.
You can continue.
Okay. So these are all about biofertilizers. I will tell you another thing, because Maharashtra has a very large coastline, right from after Gujarat till Goa. You have a lot of big coastline, though you are away from coast, but you have a coastline. Odisha has a coastline also. Odisha has a lake called Chilka Lake. It's the largest brackish water lagoon in Asia. It is connected to the Bay of Bengal and seawater influx is here. The largest in Asia. A lot of birds come. It is a good tourist site. It is 20 kilometer wide, 60 kilometer long, and it's a various salinity gradient. In this, we get a lot of seaweeds. Seaweeds are macroalgae. Mostly you get three types of seaweeds. One is Gracilaria, one is Enteromorpha, one is Chytomorpha. And these seaweeds, you can take it and dry. And these dried seaweeds, this is a Gracilaria red alga from which you can produce agar-agar. Also, you can produce seaweed liquid fertilizer. We found that there are two types of seaweeds. Gracilaria occur in Chilka Lake. What is a long variety, is a bushy variety. Quality-wise, both are okay. And what we did, we took these seaweeds, extracted the liquid extract, and made a foliar spray. Take the seaweed, dry it, put water, extract with autoclave, filter it, and use a foliar spray. It is called a seaweed liquid fertilizer. It is not a biofertilizer. It is an organic manure or organic fertilizer. This is the basic difference. Biofertilizers are always microbes. Whatever till now I was telling, those are biofertilizers. But this is an organic fertilizer or is a biostimulant. If you put these things in Vigna radiata, you can see the growth in 60 days, how it improves. We have tried with Vigna radiata, we tried with the brinjal, the SLF, and works very well. Chilka, we have checked how much of seaweeds occur. We went in a boat and artificially divided to 24 sectors, different salinity gradients. We went with a GPS and collected seaweeds. This is a Karizai island, beautiful one. I went with my students many times. I found you find the red seaweed grow inside water. This is a red seaweed, this is a green seaweed. We collect the seaweed in plenty, either from boat in a quadrant or manually to the village nearby. It has around 50 kilometers or so. These are the seaweeds. This is Gracilaria, this is Enteromorpha, this is Chytomorpha. This is Chytomorpha. three types of seaweeds we get in plenty, segregate them separately. Okay, after segregation, we develop a seaweed technology, 30 minutes at a pressure. Then cool the content and filter in a cloth, you get 100% seaweed extract. You make it diluted to 1% under hot condition and put in refrigerator for further use. Like this. Take the seaweed, separate them, dry under shade, then pulverize them, grind them in a grinder, autoclave, put it in a water 1.5 gram in 100 milliliter, or 1.5 gram, sorry, 1.5 gram in 1 liter. Then autoclave them, then filter, you get 100% liquid extract. Make it 1% and store. And this one you can use as a foliar spray, or you can use it as a seed treatment. Mix the seed and plant, or use as a foliar spray, and you are assured of 25% yield. This also we have calculated. One-time investment, we need an autoclave, glass double distillation unit, hot air oven, compound microscope for checking purity. And cost is plastic polypropylene bottles of 1 liter capacity, preservative, some sodium hypochlorite, labor transport and cost from Silica Lake, and glassware, plastic weights, etc. So total recurring expenses will be 1,52,000 per year, all these things. So if you, and these are used as a foliar spray, 1% only. So you can get selling cost of 800 liter of a cell per month you can have. And if it can be sold at 100 rupees per bottle, 80,000 rupees per month in a year, or in a year, 9,60,000 rupees per year. You invest, deduct the interest against this money, against 75,000 with a fixed interest of 15%. Now it is 5%, but 15% say. So minus this per year, variable cost 1,50,000, indirect cost, salary, wages, etc. 2,44,000, advertisement, print media, etc., 50,000. Say you will spend 4,57,000 rupees per year, and net return will be 5,03,000 per year, or 40,000 rupees per month net profit. That means a cottage industry can be done near the lake, or in other places. Transport cost you have to deduce to that. For example, in Maharashtra, anybody can do it in Ratnagiri, in Mumbai coast, or a lot of coastline of Maharashtra. They can have this seaweed liquid fertilizer industry used for plantation crop and vegetable crops. This is not a biofertilizer, but this is an organic biostimulant, can be used for quality fertilization of agriculture crops. This also I am giving training, seaweed liquid fertilizer. While I was working on this, I was not working in Bhubaneswar. I was working in Shantiniketan. That was a central university by Rabindranath Tagore. I worked there 10 years as the head of the Department of Biotechnology. There I had a person from agriculture department. Tagore started agriculture subject also in Shantiniketan. So we together worked in Sundarbans as well as Odisha coast, and both the places we made a lot of work about this seaweed liquid fertilizer, which many people now produce in Bengal as well as in Odisha. Many times, many training programs we do on seaweed liquid fertilizers in West Bengal, in Shantiniketan as well as Odisha, and it is very, very successful. And in my village, small village, wherever there is a community program, my students used to go and display the seaweed liquid fertilizer, organic fertilizers for the public. And this boy who did his PhD on this, now he's in China as a postdoc, and he is a professor, assistant professor in IIT Kharagpur right now. And these are all the village people. Just they are with us whenever there is a program going on. I will now close. So biofertilizers and green manures are very well known to be used in agriculture to combat the ill effect of chemical fertilizers. This everybody knows. Our principal Saab also told a lot of things about this.
Very informative session, sir.
So
Thank you.
let me complete. So biofertilizers and green manures are very, very important. Okay. Biofertilizers are microbes. Green manures, you can put vermicompost or compost or SLF, sesbania, all. But both the things are used to combat the ill effect of chemical fertilizers. The problem is, if you use biofertilizer or green manure, you cannot get that yield what you get with chemical fertilizers. Because chemical fertilizers, you put it, immediately you get higher yield. But if you put biofertilizer, green manures, yield is always not higher. Okay, that is the only bottleneck. And farmers, those who do farming for money, they cannot be convinced with quality. Though abroad, Germany, Japan, US, everybody, they are conscious with quality. In India, we are not farmers are not concerned with quality. They are interested in money. So that's why they are used chemical fertilizers. And biofertilizer green manures are cheaper. And with this, if the companies or the vendors will sell them, they won't get much profit. That's why they always tell the farmers to buy pesticides and chemical fertilizers. But you have to have yield, otherwise farmers won't use these things. So what we always tell, you make an integrated nutrition system. Use biofertilizer, little. Use organic manures, little. Use chemical fertilizers also. Suppose you use 60 kg nitrogen, urea. You reduce to 30 kg. And then use biofertilizer green manure so that the quality of the microbes won't be lost and quality of the soil will be little better. If you can afford, use full biofertilizer green manure. If you cannot afford, but if you cannot convince a farmer, tell him to use an integrated nutrition supply system by means of judicious combination of fertilizer, organic manure, and biofertilizers for increasing productivity and maintaining the soil health of farmland. This is my final message. Thank you very much. I enjoyed speaking to you in an accredited college under RTM Nagpur University. Thank you very much. I thank the principal, IQC coordinator, Madam Qureshi, and head of the department, all for this, hearing me for such last 2 hours. Thank you very much.
Thank you very much, sir. Very, very informative session. We gain much knowledge about biofertilizer. Thank you so much, sir.
Thank you.
Thank you, sir, for sharing your profound knowledge related to biofertilizers with us. Your session is very interesting and you share lots of valuable information. The ideas shared by you regarding organic farming, entrepreneurship, and sustainable agriculture development in India will help everyone who have attended this seminar, sir. Thank you very much, sir, once again. So we go for question answer session.
Yes.
There is some questions. I read one by one.
Yes.
My first question is, sir, which biofertilizer is good for paddy cultivation is asked by Dr. DJ Tiwari, Department of Botany, J.M. Patel College, Bhandara.
It is blue-green algae and azolla.
My question, I repeat.
Yeah, I understand. It is only blue-green algae and azolla biofertilizer.
So blue-green algae?
And azolla biofertilizer. Either blue-green algae biofertilizer or azolla.
Okay, yes.
Because they grow in water. And rice needs water.
Okay, sir. Thank you very much. My next question is asked by a student, Pallavi Bante. What are the application of biofertilizers? What are the application of biofertilizers?
Biofertilizers are applied 3 ways. Either as a seed coating, or dipping in the seedlings, or amended to the soil. But these are only for Rhizobium, Azotobacter, Azospirillum, not for BGA and azolla. Okay, and SLF, if you want to use, use as a foliar spray.
Okay, sir. Thank you very much. There is one more question asked by the student of MSc Botany, Triptil Mein. The question is, sir, what are the factors affecting the efficiency of biofertilizers?
You look, factors means these are biological organisms. Any microorganisms won't survive if the temperature is more than 35-40 degrees Celsius. If temperature is too low, they cannot grow. So there is a growth condition of the microorganism is required. Unless that condition is met, unless that agroclimatic regime condition is maintained, they won't work. Okay.
Sir, thank you. Sir, one more question is there. This question is asked by Dr. Veena Mahajan, Department of Zoology, J.M. Patel College. She asked, how do we calculate the agronomic efficiency of biofertilizers?
Well, agronomic efficiency means how much input you give, what amount of money you spend for production, how much you get the yield, what is the cost, calculate the cost economics. Okay, and about this agronomic importance, you see how much of hormones, vitamins, nitrogen they give to the crop, this soil. That has to be calculated. This is scientific method required.
Okay, sir. Thank you. Sir, my last question is, again, this question is asked by Dr. Tiwari, sir, Vijay Tiwari, Department of Botany, whether private companies are investing for manufacturing of biofertilizer in India?
There are several companies, several. I told you we have Government of India started regional biofertilizer development center. There are 4 in India. One is in Nagpur. One is in Nagpur, Central India. One is in Pune. One is in Bhubaneswar. One is in Manipur, and another one is in Lucknow, the center. Okay, so technology is available there, but problem is though they. I have told. I have given a slide about the manufacturer's concern. Okay, unless they because it's a bio, only quality is maintained, it won't work. Manufacturers should be honest, they should be serious, they must do R&D, they must employ right people, they must give good salary to the person they are employing, and to do good extension. It has to go to the farmers, they should do extensive extension programs, then only it will be successful. Simply sitting and putting some boards in Nagpur city, it won't do. Okay, one has to go to the farmers, demonstrate, train them, show them. And once this technology is accepted, one can become a millionaire. Otherwise, they will become bankrupt. Nobody will buy those things. Farmers are very clever nowadays. They see whether really they are getting the benefit or not. It's okay?
Thank you, sir, for answering all the questions. Yes, sir. Thank you, sir, for answering all the questions. Now I call upon Dr. Padmavati Rao, Head Department of Botany, to propose vote of thanks.
Namaste, sir. Good afternoon to everyone. Respected principal of our college, Dr. Vikas Domne, sir, the resource person of the national seminar, Honorable Dr. Prasad Adhikari, delegates, and my dear students. It's my proud privilege to propose the vote of thanks of this national seminar. Words are not enough to express our sincere gratitude to our esteemed resource person, Honorable Dr. Shiv Prasad Adhikari. To the best of my knowledge, he was actually traveling when we approached him with the invitation, and he kindly accepted to speak on this important note or topic. Sir, on the basis of the feedback received from the participants, I can tell you it was a wonderful lecture and delivered it is in very lucid manner. Thank you very much, sir. I would like to thank our dynamic and beloved principal, Dr. Vikas Domne, sir, for all his encouragement and support in the organization of this national seminar. Thank you very much, sir. I am also thankful to IQSC coordinator of our college, Dr. Karthik Panikar sir, and all the IQAC members for their help in the organization of this seminar. I would like to thank Dr. S.P. Qureshi for organizing this webinar, and special thanks to Dr. Aparna Yadav for conducting the proceedings. I am also thankful to Dr. V. Tiwari, Dr. Aparna Yadav, Dr. G.B. Tiwari, and Dr. Sunil Janje, Dr. Samina Tadvi for taking care of different aspects of the seminar. It has been a great teamwork. Special thanks to Dr. Romy Bist and Professor Matlan sir for their technical support. Last but not the least, I would like to thank all delegates of different parts of the country, including researchers and students of our college, for making this event successful. And lastly, I'm giving some instructions to participants. We will be posting the feedback form in the Telegram group. Fill the form correctly so that we can send you the certificates the earliest. Once again, I'm thankful to all the participants. Thank you very much, sir. Over to Dr. Karthik Panikar, sir. Over to Dr.
Thank you. Thank you, madam. Honorable Adhikari, sir, it was great listening to you.
Thank you.
I mean, I am also not a man of biology background, but I could get, since I'm interested in organic farming to some extent, I could understand some of the concepts. Some of my friends are actually into, big time into organic farming. They also, one of my friends is also a manufacturer of biofertilizers. So I keep visiting him frequently, so I could understand some of the things that you were talking about. It was explained in a very simple language. It was a privilege having you, sir. And on behalf of the college, I thank you once again. Hope we'll have you once again after the situation improves. We'd like to have you on our campus sometime.
Thank you. I will.
Thank you, sir. Thank you very much. With the kind permission of the chair, I close the seminar here. Once again, we request all the delegates, all the participants, including the student participants, to fill up the feedback form correctly. The feedback is very important for us when we plan for the next seminars. So, and to issue you correct certificates also. So please fill the information correctly. Thank you so much for joining us. It was great participating in this seminar.
Thank you.
Sir.
Thank you. Thank you.